Hello, everyone. Today we're going to cover Chapter 3, the microscopic anatomy of the nervous system. We're going to talk about the neurons and glia, and then the internal anatomy of the neuron. And then we're going to talk about genetics as well, which is a hot topic right now and is a useful thing to cover from other classes. And we'll talk about how genes affect cells and affect our behavior throughout the class. So we'll talk about these three topics today, starting with the general overview of the cells of our nervous system. So about 120 years ago, about the time that the first houses in Edmonton were being built, there was an intense debate being had between two anatomists, physiologists, Golgi and Cahal, who were starting to look through neural tissue through microscopes. And Golgi first developed a stain that allowed him to look at neurons under the microscope and observe this interconnected network, which he termed a nerve network. And he believed that all these neurons were connected together, and this is how they communicated information. Cahal, using the same techniques that Golgi developed, identified what looked like discrete cells in the nervous system, just like in the rest of our body, such that each neuron was contained in and of itself and was discrete from the neurons next to it. And this led to the idea of neurons being the functional unit of the brain, which has turned out to be true. So Cahal used this Golgi stain to show that each cell was discrete. As you can see here in a modern Golgi stain image, individual neurons and their processes, all these long tree-like structures, are discrete from one another. So a neuron, as you've learned in your previous classes, is a cell with a cell body like our other cells, but it has these often long branching structures associated with its cell membrane called either dendrites or axons. And these dendrites collect information from other cells, other neurons, and the main root axon, or axle in Greek, carries messages to other neurons in turn. So these incoming neurons' axons activate the dendrites and in turn send information down their axons to other dendrites. And most neurons have only a single axon, but many dendrites. And any one neuron doesn't contribute much to our behavior. It's large groups of hundreds or thousands of neurons that get together in functional groups or neural networks. And these connect large areas of the brain together with our body as well. And so one of the aims of neuroscience and psychology is to map the structural connectivity of the human brain. How is each part of the human brain wired to each other part? We can call this the connectome, just like the genome. The connectome is the entire connection map of the human brain, of which we've heard there are 86 billion neurons. So as we've seen, the cell can be divided into its dendrites, its cell body, or its axons, and we saw that the dendrites gather information from the previous neurons. In the chain of information, the cell body integrates this information from the various parts of the dendrites together, and the axon carries information on to other cells. Here's a more detailed look at the anatomy of the neuron under a couple microscope images, in which you can see the dendrites actually have these protrusions called dendritic spines, which end up reaching out and making the connection with the axons of the previous neuron. And so this drastically increases the surface area of the cell. You can also see an enlargement of the nucleus of the cell inside the cell body, and the nucleolus inside, where we know that the genetic material is stored. The axon is connected to the cell body in an area called the axon hillock here. So these dendritic spines, as we saw, are these small protrusions from the dendrites that increase the surface area and are the usual points where it connects to other axons. The axon hillock is the junction between the soma or cell body and the axon, where the action potentials, which we'll learn about next week, begin. The action potentials are the electrical messages that the neurons are sending. There's axon collaterals, which are branches of the axon we see on this previous slide here. And terminal buttons are knobs at the tips of axons, which make the connections to the dendritic spines. So you can see on the ends of each of these axons are little end feet or terminal buttons, which are creating these connections with the dendritic spines. And it's this area between the end feet and the dendritic spine, which is called the synapse, cavity of space extracellularly outside of either of these cells through which they transmit information, which we'll also learn about next week. So the synapse is a gap between one neuron and the other, and it's usually the terminal button of the axon connecting to the dendritic spine of the next neuron. And so information flows from the dendrites to the cell bodies, to the axons, to the terminal button, and then back to the dendrites. So just to belabor the point, the dendrites collect information from other neurons. They're processed in the cell body and then passed down the axon and sent to the next neurons, dendrites. Neurons in general can be divided into three most general types: sensory neurons, interneurons, and motor neurons. Sensory neurons are the first input into the nervous system. They carry information from the sensory receptors all over our body and our eyes and our mouth and ears into the spinal cord. The interneurons associate these various sensory informations together in more and more complicated ways and create motor plans using these associations. And then finally, the motor neurons start in our brain and travel down our spinal cord to our muscles to output commands to move our voice and our body. So most of our brain is then interneurons, are these in-between computational devices, in between the incoming sensory information and the outgoing motor information. So in general, sensory neurons collect information from their dendrites and pass it down through their axons. Here's two examples: a bipolar neuron, which we'll learn about in the chapter on vision in the second midterm, and a somatosensory neuron. The bipolar neuron has just a tiny dendrite and a tiny axon. The somatosensory neuron, which we already learned about a little bit with respect to the spinal cord last week, in which these incoming somatosensory information from our body enter the back of the spinal cord. We saw that there's a small section along the way where their cell bodies are, and their dendrites are actually connected directly to their axons. This is the part that can get squished when your disc protrudes when you have a bad back. Interneurons come in many different shapes and sizes. Here are three examples: stellate cells in the thalamus. You can see how this structure would be very good for integrating information from all the local neurons. The pyramidal cells in the cortex, which have these large dendrites and long axons, and Purkinje cells in the cerebellum, which have these gigantic arborizations or tree-like dendritic webs in the cerebellum. Motor neurons, thirdly, are generally very large, the largest neurons in our body, and they command the muscles. So they send axons down our spinal cord. You can see these axons have little insulation on them, which we'll learn about, called myelin, which transmits the information faster to our body. I talked about most of these things already, but we'll just recover it one more time. The bipolar neurons are what we'll learn about in the chapter on vision. What take information from the retina's light receptors and carry it into the brain visual activity centers, and the somatosensory dendrites connect directly from the dendrite to the axon, and the cell body sits outside. We've already talked about these three types of cells as well. So the motor neurons reside down in the lower parts of the brainstem that send their axons all the way down to the muscles of your body. And everything going out to your body must pass through them. So these sensory neurons collect the incoming information from the body, and they connect to interneurons, which process it, and they pass it on to your motor neurons. And these motor neurons move our muscles and produce behavior. So any given neural network, any group of neurons has an input, associates those inputs together, and it creates an output. And even our artificial neural networks, for instance, that look at pictures on the internet and identify who or what is in them, have these same properties. These neurons communicate with one another down their axons through a language of excitation and inhibition, and that each neuron receives thousands of incoming signals that say it should fire more, and it receives thousands that say it should fire less every second, and it sums these signals together, and it creates essentially a vote, all or none vote, whether it should send a signal itself. So this simple yes or no signal then gets merged with thousands of other signals in the next neuron. And this simple language of yes, no of each neuron, and then summing together the votes in the next level, can lead to enormous possibilities in our behavioral repertoire. A very simple way of thinking about the ability of such simple connections between neurons to create behavior is to create models of very simple animal behaviors. And for instance, roboticists can try to create simple connections between sensors on the robot and the motor effectors on the robot to move its wheels that create kind of artificial-like brain connections to identify whether our theories about how simple neural connections could lead to these complex behaviors are on the right track. So you can create this robot with microphones that detect noise and make the robot move towards that noise to replicate a male cricket seeking a mate. And we can say that the louder the sound is, the more the motor should move, and so the faster the animal will move towards that sound. So you could think about a very simple circuit with two microphones, each having an excitatory connection, or they would increase the speed of the motor. And the cricket being closer to one of the microphones will create a louder signal at that microphone, which will move this motor a little more, turning the wheel towards the cricket. So the sensory and motor neurons can summate this excitatory signals to create what looks like an intelligent being. And you could add an even extra bit of inhibition into this circuit by understanding that the crickets also avoid very bright places, and they want to only go towards their mates in the dark. And so you add light sensors as well, like eyes for the cricket, that inhibit the connection between the contralateral microphone and the motor. That is the microphone on the opposite side and the motor on this side. So if there is light on this side of the animal and the cricket is making a sound on this side, the light will inhibit the wheel on this side and will increase the wheel on this side by the microphone, and the animal will turn that way. However, if there's a bright light in the same place, it can inhibit that circuit and not allow the cricket to go. I might have mixed that up. So here, the positive connection between a microphone that usually would have turned the wheel towards it can be inhibited by the photoreceptor on the other side, which would have more light. That is, if there's light on the same side as the cricket, then the louder microphone, the quieter microphone will be inhibited even more and will move less. So it will always move towards brighter areas with sounds. Yes, I was right the first time. Okay. Moving on, we're going to think about some other types of cells in the nervous system. In addition to the 86 billion neurons, there's also 87 billion glial cells, which we think of as helper cells. Some of them that you might have heard about are astrocytes or oligodendrocytes. These have many different functions in the nervous system, which we'll talk about in turn for each one. This is just a table that summarizes the things that we'll talk about on the next slides. So ependymal cells are small and they're ovular, they're ovoid, and they're first found in the walls of ventricles. And they get secreted and made in the cerebral spinal fluid. And they seem to be involved in the cleanup of waste. Astrocytes are these large star-shaped cells that are symmetrical. They've been thought to structurally support neurons by holding a scaffolding between them. And they also transport substances between neurons and the blood vessels and help create the blood-brain barrier. When tissue damage happens in the brain, they help form scar tissue. And they also help provide the fuel for the brain, something my wife Claire studies. The astrocytes help store energy that the neurons can use later when they need it. So here's an astrocyte and a neuron showing their kind of tight relationship. The astrocyte wraps its legs around the neuron's processes, holding it in place. It also wraps its legs and arms around nearby blood vessels, holding the three together and solidifying the connections in the blood vessels, creating these tight junctions, which hold in much of the substances in our blood and don't allow them to pass into the brain. So when you've heard about the blood-brain barrier, it's happening right here in these blood vessels. Microglia, go back and look at the picture, are small. And they start in the blood, not in the brain, and they're from our immune system. So they go through our brain and they phagocytosize. They take up dead tissue and debris. In general, the blood-brain barrier doesn't allow most immune system cells into the brain, which can lead to some problems when we need to treat brain infections. These microglia, however, monitor the health of brain tissue and they play a role in its immune system. So one thing they do is attack foreign tissue. Microglia also, like astrocytes, invade areas that are damaged and provide growth factors to help grow the damaged tissue. I think I already covered that. Extra slide. Yeah. That was just the same thing as on the previous slides. Oligodendrocytes are glial cells inside the central nervous system that wrap their processes around axons, creating the myelin, which I said was insulating the axons and allowing them to conduct information faster. And Schwann cells have the same function, but in the peripheral nervous system. Well, let's just look at those pictures. So here you can see the oligodendrocytes wrapped around multiple axons in the central nervous system with their cell body outside. The Schwann cells out in the peripheral nervous system, however, have their cell bodies themselves wrapped around the axons, forming the myelin sheath. This fatty tissue, which appears white under the microscope, which is what makes up our white matter, helps conduct electricity down these axons more quickly. So this glial coating that surrounds axons, multiple sclerosis is a nervous system disorder where this myelin breaks down and therefore the conduction of information through the nervous system is delayed. Paralysis can occur due to damage of the nervous system, but when these axons are severed, for instance, in your wrist or arm, you can get a severing of some axons and lose sensation and movement in all the downstream areas. If this happens in the peripheral nervous system, the microglia and the Schwann cells can repair the neurons, and the axon can regrow and connect back to your hand. However, if it happens in the spine or the brain, this repair doesn't take place. So a spine or a brain injury can't be recovered from in the same way. This regrowth, in fact, might even be inhibited in the spine, leading to difficulty treating people that have had spinal injuries. When the neurons do repair in our arms or legs, they do it in this interesting way where the Schwann cells degenerate but leave this kind of breadcrumb path for the axon to find when it regrows. And as it regrows, it puts out many processes in many different directions, and the ones that end up down this route of the Schwann cells end up connecting to the downstream connection that they were trying to reach, leading to a reconnecting of the axon and a new function in the arm or leg. Okay, just going to take a pause here. Next, we'll zoom into the internal structures of the cell and cover the cell's features in more detail, which we will realize leads us to a discussion of the creation of proteins, how they're made, which will lead us to a discussion of genes and mutations in genetics. So generally, a cell's proteins are what determine its characteristics and functions. So what differentiates our different cells in our body are the various proteins that they've created, for instance, to be a fingernail or a finger or a piece of hair or an eyeball. Each cell can use its genetic code to generate thousands and thousands of different proteins, and each of these proteins can even be combined in different ways. Water, salts, and ions play prominent parts in the cell's function, so we'll talk briefly about the chemistry of these three molecules and cover them again. So when we open up the cell and look inside, the cell of a neuron looks much like the many other cells that we've learned about in our biology classes. It has a nucleus, a nuclear membrane surrounding the nucleus. It has an endoplasmic reticulum around the nuclear membrane in which the proteins are created. It has a Golgi body in which the proteins are packaged up for transportation. It has mitochondria, which create the energy that the cell needs to do its functions. It also has these microtubules and microfilaments, which make up its cellular structure and also a pathway for these molecules to move throughout the internal parts of the cell. Lysosomes are these little sacs inside the cell that can move these proteins around, usually along these microtubules. We should see also that the inside of the cell is separated from the outside of the cell by a membrane, and therefore the fluid on the inside of the cell and the fluid on the outside of the cell are separate. We know also that the nucleus on the inside of the cell is where the chromosomes and genes are contained. Here we can see the dendrites and dendritic spines above the cell body, and the axon, hillock and axon here, and a little bit of myelin coming out from the bottom of the cell body. So the cell membrane itself separates, as we said, the intra and extracellular fluid. It regulates the movements of substances in and out of the cell. Almost all substances cannot pass between the inside and the outside of the cell. There are some proteins that are embedded within the membrane which allow some things to go in and out, which we'll talk about today, called channels or gates. The cell membrane is made up of phospholipids that have a hydrophilic head and a hydrophobic tail. So their head is attracted to water because of phosphorus groups that have positive and negative charges, and their tail has no positive and negative charges. It's a fat molecule, and it's therefore hydrophobic, and it doesn't want to be near the water, which creates this this composition where there's two layers of these cells called a lipid bilayer, a phospholipid bilayer, which separates the extra and intracellular fluid, and nothing can pass between them because fluid is repelled from this space. Here's what the phospholipid molecule looks like in a diagram, where you can see the positive and negative charge, but there's no charge on the tails. And here's a space-filling model showing the molecular structure of the phospholipids, showing these phosphate groups with positive and negative charges. We'll talk in the next slides about some chemistry terms like binding sites here. Basically, there's no free electron spots in any of these molecules for any other molecules to bind to, but on the top there is, and water can be attracted to those areas. So this bilayer membrane separates the intra and extracellular fluid. So just to give us a brief overview of chemistry, myself included, an element, or what we see in these chemistry charts, or any of these naturally occurring substances that can't be further divided into other substances. And an atom is the smallest quantity of one of those elements that keeps its properties. And each of those atoms contains a nucleus. And in the nucleus are neutrons and protons, neutrons having a neutral charge and protons having a positive charge. And then electrons orbit the nucleus and they have negative charges. And usually the negative charges in the orbit and the positive charges in the proton balance each other out. But when they don't, ions are created, and this is where interesting chemistry can happen. So when multiple atoms bind together, molecules are created. And so, for instance, with salt, like sodium chloride, a molecule is the smallest unit of a substance that still has both the sodium and the chloride. It's not divided further, and it can't still be salt. So when sodium chloride is formed, sodium gives up one of its electrons and gives it to chloride, which is missing an electron, and they combine and are attracted together and create a crystal. And these positive and negatively charged ions held tight together by this electrical connection creates this new molecule. Water is another example of a molecule in which atoms are held together, in this case by electrons that are shared between the oxygen and the hydrogen. And because the electrons take a little bit more time to travel around the oxygen's protons and nucleus than they do around the hydrogen's protons and nucleus, they spend a little more time down by the oxygen and create a negativity on the bottom and a positivity on the top of each water molecule. So each water molecule has a charge on either end, like a small battery, and allows it to be attracted to other substances. And this property allows it, for instance, to dissolve salt into salt water, which is what comprises most of our brain tissue. So the nucleus of the cell is where the genetic code that creates our proteins is held. And this also holds all the information needed to make our brain and our body and our cells. Each cell has an identical set of genes in its nucleus that can be used by that cell in different ways. Inside the nucleus are chromosomes, of which we have 23 pairs. And each of these chromosomes is a strand of DNA, of deoxyribonucleic acid. And so this DNA is what holds the entire genetic code to recreate us. And you can even extract it from your own saliva. You can Google how to extract my own DNA, and you put in a little dish soap, I think, and you can lyse the cells and pull out the DNA, and it looks like kind of a little slimy white substance. And you can do it to fruit and to vegetables as well. And this DNA is comprised of 4 nucleotide bases, 4 little substances: adenine, thymine, guanine, and cytosine in some sort of order. And the particular order of these nucleotide bases is what represents the code, the gene itself. So a gene is a segment of DNA that encodes the synthesis of a particular protein. It's a code for how to make a particular protein in the endoplasmic reticulum. And the sequence of adenine, thymine, guanine determines which amino acids are going to make up the protein once it's created. Okay, so here's a picture of the chromosome inside the nucleus of a cell. It's this long strand of DNA. DNA is this long double helix of these nucleotide bases connected together and held together tightly. And adenine always binds with thymine, and guanine always binds with cytosine. So the code on one side is analogous and kind of complementary to the code on the other. So once information, which we'll see, leaves the nucleus, it goes into the endoplasmic reticulum where proteins are actually created. And so ribosomes take the information from well, we skipped a step here, it wasn't on the slide, but the gene, when it wants to be turned into a protein, can unfold itself from the strand like this, and then nucleotides can attach to those nucleotide bases to create kind of a complementary set of nucleotides that would match what would have been on the complementary strand of the DNA. And this is called mRNA, messenger RNA. And this messenger RNA then travels from the nucleus into the endoplasmic reticulum. And this is the blueprint for how to create the protein. It's been created from the genome, and now it's traveling into the endoplasmic reticulum where the ribosomes are going to use it to create amino acids. So the ribosomes are going to move down the mRNA, and they're going to read each of those nucleotide bases in the mRNA. And for groups of each 3 of them, they're going to create corresponding amino acid. Go back to the previous slide. So these ribosomes are themselves proteins, and they are catalysts for protein synthesis as this mRNA comes into the endoplasmic reticulum. Translation is one of the later phases of protein synthesis in which these messenger RNAs travel from the nucleus to the endoplasmic reticulum, and they're translated into the amino acids to form a protein. Transcription, on the other hand, is a very early phase, is the earlier phase in which the DNA unwinds and this strand of messenger RNA is produced that creates the code. Each 3 nucleotide bases in the mRNA create what's called a codon, and each of those codons codes for a particular amino acid. And so this might be A, C, G, and then that would make this particular amino acid, maybe it's tryptophan, and then the next 3 might code for another one. So this entire set of codons would then code for this set of amino acids, and that amino acids would be the protein. Okay, so we have this template strand of DNA, and this gets turned into the mRNA. And each 3 sections of the mRNA are a codon which translates into, so that transcribes into mRNA, and the mRNA translates into the amino acid, a chain of polypeptides. And these polypeptides in this particular order create some particular protein of use to the cell. An amino acid is a molecule with a central carbon atom and has an amino group on one side and a carboxyl group on the other that create the chain. So each amino group attaches to the negative carboxyl group of the previous part of the chain. And it also has a side chain, or R, which we can see on this slide here. So it has the carbon attached to a hydrogen, it has the amino group and the carboxyl group to connect to its neighbors, and then it has this side group R, and that's what is different from amino acid to amino acid to make them have different properties. So some things, one thing we hear about in this chapter is what's called an enzyme. An enzyme is a particular protein catalyst that makes the cell's chemical reactions more effective. Okay, so this is the chemical composition of one of these amino acids, and this is a polypeptide chain of these amino acids that together forms a protein. One example of a protein that's very popular right now in the media is the that can be made by our genetic code. By a genetic code is the spike protein on the outside of the coronavirus. And so you've heard about the mRNA vaccine. The mRNA vaccine injects these mRNA strands with the particular set of codons needed to make the spike protein polypeptide chain. And they are injected into just some of the cells in the local area where you get the injection. Those cells would then express that spike protein, that it would be created in their endoplasmic reticulum. It would be transported to the cell membrane and put into the membrane of the cell, at which time your immune cells could identify it. The virus has nothing to do with that. It's just the outside of the cell, and your immune cells would then learn to identify those spike proteins and start to create antibodies that can target them and fend them off. So the primary structure of a protein, you'll remember back from biology, is this chain of amino acids, which in the secondary structure gets pleated or wrapped like a helix. In the tertiary structure, it gets folded together and creates its protein form. And together with other proteins can even create a quaternary structure. This would be what we think of as the spike protein, this kind of mass of the proteins together. So when the endoplasmic reticulum creates a particular protein, it then enters the Golgi bodies, where they're wrapped in a membrane and given a location where they need to go. For instance, to the cell wall or inside of the axon terminal. And the small vesicles filled with those proteins can then travel down the microtubules throughout the cell and get to the destination. And at the destination, they could either be incorporated into the membrane, like these spike proteins that are created from the mRNA and the vaccine. They can remain in the cell and act as an enzyme to increase the productivity of certain reactions, or they can be excreted from the cell out into the extracellular fluid. And one reason why you might want to send these proteins into the cell wall is that they can then act as things called channels, gates, and pumps. So the proteins, all of our proteins in our body shape, and their ability to change shape depends on their sequence of amino acids. And when I say change shape, I mean that some of them can have certain chemicals or electrical charges act on them and change the orientation of the cells or their quaternary or tertiary structure to make their function different. And so if a particular stimulus, like a molecule of the right type or an electrical charge of the right amount, comes in that cell, it can open up the protein's ability to change into a different form. right? So here's an example of a protein that might have glucose bind to it, and when glucose binds to it, it kind of changes its shape a little bit. So when that shape changes, these proteins that are embedded in the cell membrane might do something interesting, like let different amounts of a molecule in and out. There's three types of openings that can occur in the cell membrane, called channels, gates, and pumps. A channel is just an opening that always allows a particular ion through. A gate only allows things through when it's open, and a pump actively moves things across the membrane. So each of these, remember, is just a protein, is a complex folded polypeptide chain. And here you can see that the potassium ion is let in through this channel freely. A gate that's open because of a particular stimulus acting on it can allow sodium through, but this particular gate doesn't let sodium through when it's closed. And here's a pump which is embedded in the cell membrane, which lets in potassium into the cell and pushes out sodium out of the cell. And it's constantly doing this over and over and over again using energy. We'll see why next week, why the cells are constantly pumping in this potassium and pumping out the sodium. Okay, so I think now we see how the internal structures of a cell give rise to proteins in the cell that support the function of the cell and support its ability to communicate to other cells. And so now we're going to look more closely at genetics and how we can use genetics and think about genetics to understand and study the brain. Because, for instance, many genetic abnormalities lead to disorders that affect our behavior and the functioning and activity of our brain by modifying the production of certain proteins that might be, for instance, embedded in the cell membrane. So we'll talk about some general terminology as well. A genotype is our genetic makeup, is what code we inherited from our parents. A phenotype is the physical expression and behavioral expression of the traits. We talked about how Mendelian genetics studies how genes influence our traits and how genes from our parents combine together to create our phenotypes. So inside the nucleus of human cells, there's 23 pairs of chromosomes, which you can see in the picture here. 1 through 22 are autosomes, and they contain pairs, one from each of your parents. And most of these contribute to our physical appearance and behavioral functions. But then the 23rd chromosomes are sex chromosomes. We can either inherit an X or a Y from our father. And these contribute both to our physical, but also to our sexual characteristics. So an allele is a copy of a gene. And so any given cell, because you have two chromosomes, one from your mother and one from your father, has two matching alleles. That has two copies of a gene. And you can either have the same one that you inherited from both of your parents or slightly different ones. And each of these copies or alleles could be either dominant, the one that's routinely expressed when you have both, or recessive, the one that's routinely unexpressed and is dominated instead by the dominant allele. If you inherit the same allele from both of your parents, then you're homozygous. If you inherited a different allele, one from your mom and one from your dad that was different, then you're heterozygous. You have both of the alleles, and you have the genetic code for something inside you that you might not be physically expressing. Genetic mutations can occur when this particular nucleotide sequence, the sequence in the DNA, has an error. So when genes are copying inside of our system, when our cells are copying, errors can arise. And even an error as small as a single change in one of the nucleotides, so that a codon, instead of being like A is ATA, can lead to one of these mutations that can have either disruptive or even beneficial effects. And these effects of one particular gene could be very specific, or they can be very widespread. Most mutations have negative effects. Two examples of allele disorders that can affect the brain are Tay-Sachs and Huntington's. Tay-Sachs is an inherited birth defects, and it's caused by the loss of the genes that you inherit from one of your parents that encode for enzymes that break down fatty substances in the brain. So 4 to 6 months after birth, the symptoms start to occur, and it results in intellectual debility, physical changes, and death by age 5. And it's caused by a recessive allele that is more common in certain populations of humans. And so if two members of those populations that don't know that they have Tay-Sachs might get together, they might get tested for Tay-Sachs disease to make sure that they don't have a very high chance of having an offspring, or sorry, a chance of having an offspring that has Tay-Sachs disease. We'll see on the coming slides what the chances would be if they're both carriers of the disease. Huntington's disease is a different type of disorder, and it's caused by a number of repeats of a particular codon on chromosome 4. So if you have too many repeats of this codon, you can create too much of this Huntington protein that can kill brain cells in the basal ganglia and the cortex. And this leads to this disorder called Huntington disease. Huntington disease, patients with Huntington disease can usually often the onset isn't until after they've had children, so they won't know prior to the time when they're planning on having children that they could be a carrier of the disease. Okay, so we'll look at the different inheritance patterns here from a recessive gene, Tay-Sachs, and a dominant gene, Huntington. So here's Tay-Sachs, in which the someone can live a healthy, normal life that has only one, they're heterozygous for the gene for Tay-Sachs, and they have a dominant gene, and they have a they have a recessive gene that carries the mutation. And if they marry someone without that recessive gene, then they can have offspring that are carriers, but they'll have no offspring that are going to be able to inherit both of the mutated alleles because their spouse doesn't have any of them. And so, for instance, when two Jewish individuals or two French Canadian individuals get together, their doctor might screen them and are planning on having a child. Their doctor might screen them for Tay-Sachs disease to give them an idea of the chances that their child will have be a carrier or will have the disorder themselves. If both parents are carriers, then they'll have a 1 in 4 chance of having a child that has the full-blown disorder. And 2 out of 4 of their offspring will be carriers, and 1 out of 4 would have both normal alleles. Okay, on the other hand, for Huntington's disease, because it's the dominant, even if a carrier mates with someone that doesn't have either of the alleles, their offspring have a 2 or 1 in 2 chance, or 2 out of 4 chance of having the disorder themselves. You only need 1 of these alleles to express the symptoms. And you can also mate with someone that is a carrier themselves as well, or might even have both of the dominant Huntington alleles, and your offspring would then have an even more likely chance of having this Huntington disorder. So these genetic disorders can result from an aberration in part or all of the chromosome. And they're different from what we just spoke about, where that was rather just a single codon or a single protein, part of the polypeptide chain that was modified. In this case, maybe the whole chromosome might be duplicated. So for instance, in Down syndrome, the mother can pass on 2 copies of chromosome 21, and paired with the father's 1 copy of chromosome 21, this can create then 3 chromosome 21s, a trisomy. And it's usually the mother, it can be the father as well. And this can create this disorder called Down syndrome, which leads to intellectual impairment and physical abnormalities as well, based off the change in proteins expressed by having 3 of those chromosomes. Finally, we're going to end this lecture on genetics and the microscopic anatomy of the cell with a talk about genetic engineering and how it relates to neuroscience. So one really important way that we can study the brain is by identifying which of these genes give rise to different neural structures or activities, and then in animal models, thinking about modifying them, or in humans, trying to identify humans with or without these genes and selectively studying them. So we can modify a gene by adding or removing it from the genome, and there's a number of ways to do this. The most classic is by selective breeding, which humans have been using for tens of thousands of years, in which you identify, and which Mendel did in his pea plants, you identify offspring that have characteristic traits that you like, and you mate them together. And when you mate these offspring together, you then increase the propensity for those traits to be in their offspring. And this has led to this amazing assortment of dog breeds and cat breeds that we see, and also different domesticated animals. And but you can see it's very different from an approach where you understand the genes themselves, because you're actually just modifying the phenotype. Things that modify the genes themselves could involve cloning. So you could take a full genetic makeup from one animal and inject it into the egg of a mother and create a perfect copy of that animal. We'll talk about transgenic techniques and knockouts as well. So I already spoke a little bit about selective breeding. You can get these things, for instance, you could breed a whole colony of mice and wait for a spontaneous mutation to occur, like a particular color that you like, or whisker size, or speed, and then you can start to breed those animals. And unfortunately, these kind of ideas led to some terrible parts of our human and Canadian history in the eugenic movement, in which humans start to try to understand how selective breeding of humans could be used, and this led to disastrous consequences even here in Edmonton. So on the other hand, cloning is producing an offspring that's genetically identical to the animal. So you can create a perfect clone, and this is useful if you want to try to control things in your experiments. If you take 100 rats from the streets of New York and you put them in your experiment, each one of them is going to have very different genetic history. But if you take a perfect cloned rat from a mother rat, and you put 40 of them that are identical clones into your experiment, you can see how much more control you now have. This will allow you to study what influences heredity has separate from the environment, because if any of those cloned rats have different expression in their phenotype, you know that those were environmental influences, given that their genotypes were identical. This can also be used to create new organs or tissues. And there's even proposal that they can reanimate extinct animals like mastodon or even dinosaur. Okay, so we can also knock in or knock out genes in certain ways. So you can add a gene into a genome, or you can take out an inactivated gene from a genome of a mouse. one important new way that scientists are doing this knock-in or knockout technology is called CRISPR, which was developed based off some cool mechanism that bacteria has to fight off viruses, in which they can actually cut the DNA of the virus. Scientists harness that skill from the bacteria and now use it in the lab to selectively cut the beginning and end of certain genes to take them out or put them into the genome. So as we've seen, even with the same genetic material, the environment can drastically influence the phenotypic expression of these genes. So every individual around has the ability to develop into more than one person. If you exercise every day for the next 10 years, your body will express different genes than if you sit on your car and on your couch every day for the next 10 years. So phenotypic plasticity, as we've covered in the previous chapter, is the capacity for the genome to express a large number of different phenotypes. The genome stays identical, but its way of being expressed can be manipulated in different ways. Epigenetics is the study of the environment's influence on the selection of our phenotypes. So these are two cloned animals that were given different environmental influences, and one came to develop a corpus callosum and one didn't. And so the same genes can lead to different phenotypic expression based off the environmental exposure of the animal. And so another way we know that this is true is that identical twins, which share a genome in common with one another, have a lack of perfect concordance in things like breast cancer and skin cancer, showing that the environment that they grow up in has a large impact on their outcomes, not just their genes. So the environment can allow a gene to be expressed, or it can prevent its expression. This is called epigenetics. And at the most simple level, this even describes how our single genetic code can differentiate all our cells from the first cells that start in the embryo into this entire organism that we are, the 87 billion neurons and all the things that they do, is by the environment of the local cells as they divide being slightly different so that they know to turn into bone or skin or tissue. And this can also lead to disorder and to cancer as these cells divide, right? So as the environment in which they're dividing can lead to dysfunctional division, leading to cancer. So epigenetics can either block a gene or they can unlock a gene. This environment can do this. And the environment can do this through some different methods likely, and 3 of them we'll talk about in the next figure here. One is that the DNA, when it's wrapped together in the chromosome, is wrapped together very tight by these histones. And when the DNA needs to unwind to expose a gene to be transcribed into mRNA, that can be inhibited by these methyl groups attaching to the histones. And so one way the environment could influence the expression of genes is by influencing these methyl groups. The methyl groups could also attach themselves to the DNA and block the transcription process. And they can even, you can even have this RNA that blocks the mRNA being translated, and these could all be influenced by environmental factors. And one really fascinating thing that is mentioned in the textbook is that these changes in the genetic code seem to be able to be passed on down to our offspring, which is against what we were learning about in these classes 10 years ago. We learned about Mendelian versus Lamarckian genetics, and Lamarck had this idea of the giraffe evolving by every generation, stretching its neck larger and larger. That was largely kind of disproven by the Mendelian genetics, but it's coming back into flavor that the influences on your genetic system in this generation can seem to be passed on to future generations. So famine in the past of a particular colony seems to be able to, the effects of famine from years or 2 generations past seems to be passed on through to the offspring in some interesting studies mentioned in the textbook. And this is a really hot and important area for research. So that covers everything from this chapter, and in the next chapter we'll talk about the activity of neurons and how they communicate.